Welcome back everyone. Today we learn about limiting factors of photosynthesis. First let's recall what are the factors necessary for photosynthesis. Plants need carbon dioxide, which are absorbed from the atmosphere via leaves. Plants also need water that is absorbed from the soil via roots. Chlorophyll is the site of photosynthesis and located mostly in the leaves. Plants need sunlight, which is absorbed by chlorophyll. And lastly, temperature can affect photosynthesis as photosynthesis is catalyzed by enzymes. The factors highlighted in green are the three most common limiting factors of photosynthesis, and we will now look into detail how we can run certain experiments to show how they are limiting factors. There are two standard procedures done in photosynthesis experiments that one usually carries out, and that is the de-starching of plants and the testing of starch. Let's look at how this is done in greater detail. For the de-starching of plant, it is performed to ensure that the plant doesn't contain any starch so that rate of photosynthesis can be examined. The plant is kept in the dark for at least 48 hours. In the absence of photosynthesis, all the starch stored in the leaves will be converted to sugar and used up. For the testing of starch, the leaf is boiled in water for 30s to break down the cell membrane. This is to allow access to starch stored in the plant cell. Next, the leaf is soaked in alcohol and warmed gently over hot water. The alcohol dissolves and removes chlorophyll, which facilitates the testing of starch, as brown iodine added in the presence of green-colored chlorophyll can be seen to be black. The leaf is then spread on a white tile and covered with iodine solution. If iodine turns blue-black, starch is present. If iodine remains yellow, starch is absent. In all experiments, the use of controls are very useful. The control plant is to be given everything it needs, including the substance being tested during conduct of experiment. The control plant and plant to be examined on are treated in the exact same manner. But the control plant may be given the variable that is not given to the plant being tested. For example, the control plant and plant to be examined are given the same temperature and amount of sunlight, but the control plant gets to receive carbon dioxide but not the plant to be examined on. Now let's see how we can study the effect of carbon dioxide on the rate of photosynthesis. There are two experiments that can be done. In the first experiment, we will de-starch the plant and then set up the experiment as shown. Alkaline sodium hydroxide removes the acidic carbon dioxide from the air in one of the conical flask. The other that contains distilled water serves as a control. The petroleum jelly is used to smear the rubber bung to ensure that it is air-tight and no carbon dioxide in the air can diffuse into the conical flask where the leaf is sealed. After a few hours, test for starch. The lack of starch indicates the absence of photosynthesis in a carbon dioxide-free environment. The next experiment we can do is to set up the experiment as in the diagram shown below. In this experiment, we will vary the amount of carbon dioxide by adding different concentration of sodium hydrogen carbonate. The higher the concentration of sodium hydrogen carbonate, the more carbon dioxide is provided to the submerged aquatic plant. Rate of photosynthesis is then measured by counting the number of oxygen bubbles produced per minute. Now let's see how we can study the effect of light intensity on the rate of photosynthesis. There are two experiments that can be done. In the first experiment, we will de-starch the plant and then set up the experiment as shown. The portion of the leaf that is covered with the black card or aluminum foil serves to shield chlorophyll from sunlight. After a few hours, test for starch. The lack of starch at the yellow-brown area, as seen in this diagram, indicated the absence of photosynthesis where light is unavailable. Another leaf on the same plant can act as the control, and when tested for starch it would be blue-black in color. In the next experiment, set up the experiment as shown in the diagram below. We will vary the light intensity by changing the distance of light source from the setup. Similar to what we have seen previously, we will measure the rate of photosynthesis by counting the number of oxygen bubbles produced per minute. Now we will look at some graphs and interpret them. First, we have the graph of rate of photosynthesis against carbon dioxide concentration. As carbon dioxide concentration increases, the rate of photosynthesis increases, which shows that carbon dioxide is a limiting factor. However, up to a certain point the rate of photosynthesis becomes a constant as carbon dioxide is no longer the limiting factor. The rate of photosynthesis is now a constant is limited by other factors such as light intensity and temperature. Second, we have the graph of rate of photosynthesis against light intensity. As light intensity increases, the rate of photosynthesis increases, which shows that light intensity is a limiting factor. However, up to a certain point the rate of photosynthesis becomes a constant as light intensity is no longer the limiting factor. The rate of photosynthesis is now limited by other factors such as carbon dioxide concentration and temperature. Lastly, we have the graph of rate of photosynthesis against temperature. As temperature increases, the rate of photosynthesis increases until an optimum temperature. If temperature is higher than the optimum temperature, the rate of photosynthesis decreases due to denaturation of enzymes. That's all for today. Thank you for the effort to learn with me. Stay tuned for more interesting lessons. See you next time. Bye-bye.